Good day everyone, we are the reporter for this chapter which is the chapter 13, Genes Therapy. By the way, I am Emilio Baguio Jr. and now let's start the discussion on the learning objectives on this chapter. After going through this module, you must be able to describe gene therapy and its various forms, analyze an issue of gene editing employed in gene therapy, assess the problem's potential benefits and detriments to global health, explain what is genetic modified organism or GMOs and how GMO possibly affecting the society and the environment. Biotechnology. Biotechnology is the technology that utilizes biological systems, living organisms, or part of this to develop or create different products. It has tremendous application in different fields such as therapeutics, diagnostics, processed food, waste management, energy production, genetically modified crops, and many more. Technically speaking, biotechnology is the integration of natural sciences and engineering sciences in order to achieve the application of organisms, cells, and molecular analogs for products and services. The term biotechnology was first used by Carl Ehrlich in 1919, meaning the production of products from raw material with the aid of living organism. Biotech application. So there are 4 parts of biotech application. First will be the biotech application to the agriculture, which is the GM planting mats and hybrids, micropropagation, biofertilizers and biopesticides, and animal improvement. Second will be the biotech application to the environment, which is the soil and water remediation and the biosafety of GMOs. Third, the biotech application to the healthcare, which is the rNA products, vaccine and diagnostics, monoclonal antibodies, stem cells, tissue-specific. And the fourth will be the biotech application to the industry, which is the enzymes, fermentation-based products and food, biotech instruments and equipment, bioenergy and biofuel, bioinformatics. Everyone desires treatment and cure of illnesses. The recent advancement can now address this concern through biotechnology application, which is the gene therapy. So gene therapy is the technology that works in manipulating the genome to correct defective genes to treat heritable diseases. It is therapeutic strategy used engineering techniques to cure diseases or disorder caused by genetic problem. So meaning, any of the illnesses can be treated through gene therapy as long as it's related to a gene problem. Gene therapy allows the correction of disease-causing mutation to prevent the onset of diseases. It involves the process known as gene editing, which results in modified genes. Gene editing is more precise than genetic engineering, which targets a specific sequence. In gene editing, the faulty genes are corrected or edited by inserting good ones. Although the technique is a breakthrough in the medical field, some controversies occurred in using this novel technology. Genes and human genome. Genes are often called the blueprint of life that carry the information responsible for the individual traits. These are the genetic materials in our body cells, often called the DNA or the deoxyribonucleic acid. The DNA or the genes contains the instruction that directs the cell in our body to make proteins. Proteins do varied tasks in our cells. For example, it produces pigments, makes muscles strong, and kills bacteria that invades our cells. Keratin protein, for example, is produced by specific genes in the cells of our body line together to form the skin and the fingernails. Next, we have a genome. A genome is a complete set of DNA or genes in a cell or organism. It contains all the information needed by the cells in our body to build, grow, and develop. A human genome consists of DNA packed together in 46 chromosomes. These chromosomes are in homogeneous pairs. Each pair contains a paternal chromosomes from the father and a paternal chromosomes from the mother. Thus, each human cells have 23 pairs of chromosomes. 22 pairs are autosomes described as non-6 chromosomes and a pair of 6 chromosome which is X and Y chromosomes. These chromosomal pairs dictate the organism characteristic. Next, we have chromosomes. A chromosomes consists of two chromatids known as sister chromatids which contain sequence of DNA or deoxyribonucleic acid. The nucleus of the cell houses the DNA. The DNA is a double helix structure consisting of two strands. Each DNA strand is composed of many genes made up of many chemical bases such as adenine, thymine, guanine, and cytosine. Changes in the sequence of these chemical bases can result in genetic disorders.
I'm Divine C. Antolihao, the assigned reporter of the Genetic Disorders. What is genetic disorder? It's a disease that is caused by a change or mutation in an individual's DNA sequence. When we say an individual's DNA sequence, it means that it is the order of the four chemical building blocks, the carbohydrates, lipids, proteins, and nucleic acids that make up the DNA molecule. Autosomal single gene disorder occurs in individual single mutant copy of the disease-associated gene, while the X-linked disorder is a trait where a gene is located on the X chromosome. Example of this condition is the Duchenne muscular dystrophy. It is a genetic disorder characterized by progressive muscle degeneration and weakness due to the alterations of protein called dystrophin that helps keep muscle cells intact. Next is the Huntington's disease, or HD. It is a rare inherited disease that causes the progressive breakdown or degeneration of nerve cells in the brain. Also, it has a broad impact on a person's functional abilities and usually rather results in movement, thinking, and psychiatric disorders. Moreover, family members inherit this mutation from generation to generation. Next is the cystic fibrosis, or CF. It is a genetic inherited disease that causes sticky thick mucus to build up in organs, including the lungs and the pancreas. In people who have CF, or cystic fibrosis transmembrane conductance regulator, or CFTR, thick mucus clogs the airways and makes it difficult to breathe. It also includes the management ways of clearing lungs and eating correctly. Number 2. The chromosomal disorders. It is an any syndrome characterized by malformations or malfunctions in any of the body systems and caused by abnormal chromosome number or constitution. These are: first, excess or lacking chromosome, rearrangement of the chromosome, and contiguous gene syndrome. Second, structural abnormalities occur. Third, abnormal breakage and reconstruction of chromosomes results to rearrangement. Fourth, structural rearrangement lead to loss or gain of genetic material. Fifth, losing genetic materials leads to harm than gaining. Sixth, the gained genetic material leads to Down syndromes with extra chromosome total number of 21, trisomy 21, 3 copies of chromosomes instead of 2. Number 3. Multifactorial inheritance. It happens when more than one factor causes a trait or health problem, such as birth defect or chronic illness. These are the results in using cigarette or not taking diet properly. And there is a 5 to 10% possibility of multifactorial inheritance in the first degree relatives than the second degree. And the example of this is cancer.
So we're done with the genetic disorders, and now let's move forward to gene therapy and how it works. By the way, I am Lovely Mary Docindo, one of your reporters for today. Bautista et al. In 2018 described gene therapy as a method of treating or preventing diseases by replacing, repairing, or turning on or off genes of a patient's cells. Gene therapy holds promise for treating a wide range of diseases such as cancer, cystic fibrosis, heart disease, diabetes, hemophilia, and AIDS. Gene therapy focuses on correcting defective genes by, first, randomly inserting a normal gene into the genome. This is the most common technique. Second is replacing the abnormal gene with the normal gene. The third one is repairing the abnormal gene. The last one is altering regulation of a particular gene. The gene therapy method of treating conditions involves using vectors, the viral methods, or without vectors, the non-viral methods, to move DNA or genes to one cell. Conversely, vectors are vehicles or agents used to introduce or insert genes into the cell. Some gene therapy vectors include retrovirus, adenovirus, adeno-associated virus, cis and transacting elements, and herpes simplex virus. In inserting genes, it is usually has to be delivered using a carrier called a vector. The most common gene therapy vectors are viruses because they can recognize certain cells and carry genetic material into the cell's genes. Researchers remove the original disease-causing genes from the viruses, replacing them with the genes needed to stop the disease. So, let's move to the types of gene therapy. The first type of gene therapy is the somatic gene. It is the transfer of a section of DNA to any cell of the body that doesn't produce sperms or eggs. Effects of gene therapy will not be passed onto the patient's children. So, the second one is the germline gene therapy. It is the transfer of a section of DNA to cells that produce eggs or sperm. Gene therapy has its benefits and disadvantages. Let's talk about first the gene therapy benefits. So, first, it offers a cure for cancer, Parkinson's disease, AIDS, asthma, diabetes, heart diseases, and other hereditary diseases. The advantage of the technique is to give someone that is born with a genetic disease or who develops diseases the chance at a normal life. Second, it can replace defective cells. Gene therapy is used to correct defective genes in order to cure a disease or help your body better fight disease. The last one is it has promising potential in the field of medicine and other related areas like agriculture. Disadvantages of gene therapy. First, modifying organisms means modifying their capabilities. Second, viral vectors could recover their ability to cause disease. It's possible that once introduced into the body, the viruses may recover their original ability to cause disease. Possibility of causing a tumor. So, if the new genes get inserted in the wrong spot in your DNA, there is a chance that the insertion might lead to a tumor formation. Third, it can damage the gene pool resulting in multi-gene disorder. Number 4, high cost. Gene therapy can be extremely pricey, making it inaccessible for some people. And the last one is the ethical issues. The ethical issues of gene therapy. The manipulation of genes through gene therapy that changes the organism's genetic instructions raises some ethical questions or concerns. Some of these ethical questions listed by the National Library of Medicine in 2021. These are: how can good and bad uses of gene therapy be distinguished? Who decides which traits are normal and which constitute a disability or disorder? Will the high cost of gene therapy make it available only to the wealthy? Could the widespread use of gene therapy make society less accepting of people who are different? Should people be allowed to use gene therapy to enhance basic human traits such as height, intelligence, or athletic ability? Is germline gene therapy ethical?
Another application of biotechnology is genetic engineering, also called genetic modification. It refers to the deliberate modification of a characteristic of an organism by manipulating its genetic material using recombinant DNA techniques so that the capabilities of an organism become beyond what is considered normal. Scientists manipulate an organism's genes by introducing, eliminating, or rearranging specific genes which can be applied to all types of organisms including bacteria, plants, animals, and even to the human. An organism that is created or modified by genetic engineering is called a genetically modified organism or GMO. Genetic modified organism or GMOs and its controversies. Some of you will ask, what are genetic modified organism or GMOs? Are GMOs harmful? What is the purpose of creating GMOs? In connection with the different organisms on Earth, modern biotechnology produces new organisms by directly manipulating or altering the organism genome to produce genetically modified organisms with a better desirable traits through a process known as genetic engineering. Although people have practiced selective breeding to produce organisms, this modern biotechnology or the genetic engineering offers a more efficient and effective method. Some genetically modified foods according to Mania 2016 are shown in the picture. We can notice that there's a Hawaiian papaya, sweet corn, rice, and etc. Biotechnology's main goal is to provide the means to feed the hungry. The benefits of GMO in increasing crop yields have already been established. These include the enhancement in its nutritional value, resistance to certain pests, diseases, or environmental conditions. However, several studies have also reported the negative impact of GMO on health and environmental, such as their toxic effects, pesticide resistance, and ecosystem imbalance. With both the benefits and risks of using GMOs, there's an implication for long-term studies on their potential risk. Also, proper protocols must be established to ensure the safety of the people and the environment. Bautista 2018 outlined the impact of GMOs, the environmental, such as first, a direct toxic effect on non-target species by an insect-resistant GM crop. Second, an indirect effect to other species by reducing the wildlife source of foods because of GM crop. Third, pest resurgence resulted from insects that become resistant to chemical when used on pest-tolerant GM crop repeatedly. This repeated use of chemicals on GM crops may increase the number of insect pests. And fourth, an imbalanced nature created by the GM crops that change the predator-prey ratio.